Okay, we'll go ahead and get started with today's introductions. So I'd like to welcome everybody to today's webinar for World NTM Awareness Month. Today's subject is Patients, Phages, and Progress, Building a New Paradigm for NTM. I am your host and moderator, Amy Liebman, President of NTM Info and Research. Welcome, everybody. Today's guest speakers are Johan Wickstrom and Robert Hopkins, MD. Johan Wickstrom brings a deeply personal perspective to his work, having lived with Mycobacterium abscessus himself. After spending 8 years at Google working on advanced machine learning, he turned his focus to fighting antibiotic-resistant infections. As the founder and CEO of Tolka AI Therapeutics, he leads a team dedicated to developing personalized phage therapies for hard-to-treat bacterial diseases. Under his leadership, Tolka's lab made a breakthrough by discovering the first lytic phages that work against the smooth form of M. abscessus, a major step forward in offering new hope to patients with limited treatment options. Johan's journey combines cutting-edge science with lived experience, driving a mission to make safer, more effective treatments available to everyone who needs them. Dr. Hopkins is board certified in internal medicine and infectious disease and has held clinical and product development positions over 30 years at FDA, Merck, Eris, BARDA, and multiple biotech companies. Prior to Tolka AI Therapeutics, Dr. Hopkins was the Chief Medical Officer at Adaptive Phage Therapeutics and Ocugen, where he led clinical development and medical affairs activities. He has approved multiple therapeutic and prophylactic products in the infectious disease space. He is passionate about finding solutions to difficult-to-treat infections, including those due to non-tuberculous mycobacterial and infections and tuberculosis. So today, during the seminar, if you have questions, you can drop them into the Q&A box and then hit enter. If you want to send a question anonymously, you just check the send anonymously box. When the presentation is finished, we will then start reading questions, and hopefully we will have time to get through all of them. We'd like to thank our sponsors for World NTM Awareness. It is thanks to sponsors like these that we can bring this great programming to you and celebrate World NTM Awareness every year. We thank you all for your support. We'd also like to thank our partner organizations for helping us amplify the message for World NTM Awareness every year. We really appreciate their support in helping to get the message out. This year we've been doing better than ever, and we've had a lot of airway coverage and a lot, so to speak, and a lot of media coverage thanks to their amplification of our message. If you'd like to help out, go to worldntmawareness.org and you can find our media toolkit there, and you can find our social media messages, and you can help us by posting them on your social media yourself. So I'm going to go ahead, and also I wanted to remind you also worldntmday.org, and you can also sign up for the upcoming webinar on August 28th. We are going to review DPP1 inhibitors for bronchiectasis, overview and Q&A. And I know a lot of you are watching that topic very closely, as tomorrow we have a very big date coming up for an approval. And stay tuned for that. We will also keep you posted on that tomorrow. I'd like to turn it over now to Johan and Robert. Thank you so much for coming, and we look forward to hearing what you have to say.
Perfect. Thank you, Amy. I am going to kick off presentation right here. Okay, that looks like it's working. Okay, perfect. Well, thank you and welcome everyone to our talk. We've named it Patients, Phages, and Progress, Building a New Paradigm for NTM. Like Amy said, I'm Johan. I'm the CEO and co-founder of Tolka AI Therapeutics. Bob will introduce himself a little bit more later on. Everything that Amy said, I want to add just one thing on top of that. So I have a background in engineering, but I also have a background as a patients with non-tuberculous mycobacteria infection. I had Mycobacterium abscessus for 18 years. And we've always been watching the space from the sidelines, looking to see whether there are any new drugs coming down the pipeline. And a few years ago, me and my wife, who's a physician, we came across this concept of phage therapy and the very impressive progress that had been made in academia, mainly by Graham Hatfield's group at University of Pittsburgh. And we felt like this had a lot of promise, but we also felt like there wasn't enough or fast enough progress on commercializing this technology and bringing it to the market so that more people could use it. So that's why we decided to start Tolka AI Therapeutics, start our own lab isolating our own phages. And since then, we've isolated a number of lytic phages that we will talk about soon. But before we do that, let me just give a little bit of a primer why we do this at all. So pretty much everyone listening to this talk will be NTM patient or NTM adjacent. But it's worth remembering what NTM patients actually endure. Non-tuberculous mycobacteria, this data comes from a study that Amy ran actually a few years ago, where she sent out questionnaires and asked the community. And non-tuberculous mycobacteria, it's very common to have delayed diagnoses, long treatment cycles, and high symptom burdens, especially fatigue, cough, and breathlessness. And the treatments, as you all know, can be pretty brutal as well. Many side effects that come with the treatments are permanent, like, for example, tinnitus and vestibular issues. Social isolation and an unpredictable disease course impacts both mental and emotional well-being. And top treatment goals that we all share include culture conversion, less coughing, more energy, and a better quality of life. So taking all of that into account, why are the current treatments not adequate according to us? Well, taking a step back, there are two types of bacterial infections. There are the easy to treat infections, and then there are the hard to treat infections. Easy to treat infections, they make up the bulk of all bacterial infections. 99% of infections are easy to treat. This involves your common pneumonia, your skin infections. And then there's this group of the hard to treat infections that are quite rare, and this involves the NTM infections. They are rare, but they take up about half of all of our efforts and all of the money spent on treating infections, all of the work that physicians spend in treating these infections. As we all know, NTM infections are radically different to treat. And we believe that they should be treated in a radically different way as well. So non-tuberculous mycobacterial infections, they make up only 0.03% of infections in the United States, but they make up 15% of global spend on antibiotics. We use more antibiotics and more expensive antibiotics for this disease than pretty much any other bacterial disease. And that's not what most infections look like. Most infections are easy to treat. So the patient shows up at their doctor's office. They describe their symptoms to the physician. The physician will pick just one antibiotic from a list, and oftentimes this choice is pre-made. There's a guideline that says, pick this antibiotic. They prescribe it to the patient picks it up. You treat for 14 days and you stop. You don't need to check anything because most of the time, infections just resolve with one antibiotic. And we all know that NTM is not like this, but the broader market of antibacterial development, in many corners of this market, this is still the assumption that infections are easy to treat, and the only way to develop a new antibiotic is to try to treat as many infections as possible for a short period of time. Now, we believe that the future of antibacterial treatments is very different. So NTM infections are hard to treat. So instead of just describing symptoms, a patient will show up at their physician's office, and can be a multiple month process. We will take cultures, they will describe their symptoms, you will get imaging, and as you know, there can be a long period of watchful waiting as well involved, because we all know that the antibiotic treatment for non-tuberculous mycobacteria can be rather brutal. And once the decision has been made to treat, once the physician and patient has decided to treat together, they'll pick 3 antibiotics. The physician picks 3 antibiotics, or maybe 4, out of a very small list of pre-approved antibiotics. We treat for 3 months, after which we reassess and we get one culture, potentially more, but usually one. We ask about the symptoms and we decide, do we stop, do we keep going, or do we pick 3 new antibiotics because the old ones were not working or they caused severe side effects? And on we go. And eventually this treatment becomes more and more personalized and less standardized the longer we go. So very different from an easy to treat infection. So today we are already in a place where we make quote unquote personalized treatments for non-tuberculous mycobacteria. We combine antibiotics together, and most patients end up with something that maybe only they use, or at least a combination that only they use. And some issues we have with this paradigm is that we are limited to a very small set of drugs, that initial set of maybe 6 antibiotics to pick from. It becomes trial and error based very quickly. Guidelines exist for when to start treatment and how long to go. But as we all know, it is very common to deviate from the guidelines eventually. And since we gather such a small amount of information from the patients that we treat, we will also be slow to adjust if the treatment is working, or if the treatment is not working, is not obvious until you're many, many months into the treatment, potentially years into the treatment in some cases. So we believe that we need to work towards a new paradigm of treating non-tuberculous mycobacteria. We believe that precision medicine is definitely the way to go, and we need to go deeper into precision medicine and add personalization via phages and phage therapy. So this is what that could look like in the future. So we would make a tailored treatment and collect more patient data, and hopefully that will lead to better treatment outcomes. So we start with the same process where we gather cultures, we ask about symptoms. Ideally, we would ask about symptoms in the form of a patient-reported outcome, something that is more standardized and that we can store in, for example, a patient registry, so that this information can help you, but it can also help other patients to optimize their treatments. We would take imaging just like we do today. Ideally, we would also add data from wearables. So for example, I'm wearing an armband that tracks my sleep. It tracks my level of activity, my workouts, heart rate, respiratory rate, blood pressure, etc., etc. This is not a medical grade device, but it is very useful to keep track of overall health. And again, the decision to treat would be a joint decision between the physician and the patient, but ideally based on far more information than we have available today. And instead of just having antibiotics to pick from, we would still have the same list, small list of antibiotics, but we would be able to add phages to this mix from a very large library of hundreds, if not thousands, of phages. So we can make a very precise treatment just for that patient. And during this period, until we meet with the patient again and reassess the situation, we would continue to gather information from the patient. So ideally, we would take cultures more frequently. We would assess symptoms more frequently in a standardized format. And ideally, we would also get data from wearable devices. And then we, just like before, we meet with the physician. And now the physician will have a larger set of information to make those decisions together with the patient, to continue treating, to stop, or to tailor a completely new antibiotic and phage therapy mix. So the future state will look something like this. We start with rich data, cultures, symptoms, imaging, and wearables. We will create a bespoke antibiotic phage combination, either through phage hunting, where we go out and hunt for phages in the environment, which we at Tolka have been doing, and relatively successfully, or through screening of large phage libraries. We've also built a phage library at this point that is growing every month. Then provide this treatment as a nebulized treatment Uh, we would like to avoid IV therapies as much as possible, although we don't completely rule it out. And we would like to treat throughout the disease spectrum to maximize the chance of a microbiologic cure, but also a long-term clinical benefit. So we have not established yet whether it's better to treat early or treat later, but hopefully in the future we will be able to gather that information. And monitoring in real time or near real time to quickly determine if the treatment is working. We want to make sure that there's enough data available for the patient and physician to make these decisions faster if the treatment is working or if it is not working, and have real objective data to show that. That is helpful for the patient and physician. Could also be very helpful if you live in the United States and you need to show something to the insurance company to prove that, I need this medication, not that medication. So this will be sputum culture, patient-reported outcomes, cough, and potentially other measures of treatment success. So with the current 3-month check-in model, sometimes we get some of these, but ideally we would have a situation where we get more of these and more frequently for every person. That's how we can really personalize treatment for everybody. So I'm going to hand off to Bob now to talk a little bit more about how phage therapy actually works, what it is, and why it is different from antibiotics.
Okay, thanks, Johan. So I'm Bob Hopkins. I'm by background internal medicine, infectious disease physician. I do practice medicine, but I primarily do research. And my more recent experience was with about another company, about 4 years of developing phage therapy, and started about 4 different clinical trials, as well as worked on about 40 different expanded access compassionate use programs for individual patients. So I feel like I have some experience in actually getting these types of products to market. Most of my experience is in vaccines and antibiotics also, so I can do a relative comparison of what it's going to take to get a phage therapy developed as compared to what would be a typical antibiotic. In thinking about, what I would communicate today, I mean, initially I went back to actually one of the early NTM-IR conferences, and it was on phage therapy by Dr. Graham Hatfield and Kira Cohen at Johns Hopkins at University of Pittsburgh, respectively. And really, some of the actual, I couldn't really improve upon some of their presentation, their slides. They're very, very good. So I'm going to repeat and explain in a little different context exactly, what it is, how you use phage therapy, some of the history behind it, how it differs from antibiotics, define actually what a phage is. So that'll be repetitive a bit. When should you consider treating patients with phage? And how to find and or hunt for phage, and how to manufacture it. And then finally, getting into some of the clinical development, some of the safety data and efficacy data that's been generated, and what it's going to take to actually get this stuff commercialized. And then finally, we're going to end with some comments about compassionate use, because that's something we have ongoing at Tolka. So my focus is going to be more on clinical development, actually what it's going to take to get these things commercialized, but also explain how you guys can help us, and how we can help you in actually getting this product out to the market. So the first slide is just describing some of the history. This is Dr. Felix Harel. He's been credited as the father of phage therapy. He's a French-Canadian microbiologist. He actually coined the term bacteriophage in 1916, which in French literally means bacteria eater. And he treated a 12-year-old boy with dysentery and was able to cure it just with a single dose. And the next slide has an outline of how phage therapy has developed as compared to antimicrobial development. And you'll see at the bottom there, 1929 is when Alexander Fleming discovered penicillin. And you can see that from that point on, the Western world went haywire, where they just went crazy with antibiotics. Whereas in Poland, Georgia, and Russia, there was quite a bump in terms of activity and development of phage therapy. In the Western world, really, there wasn't anything going on for almost 4 decades. And then more recently, as antibiotic resistance became a problem over the decades, there became more activity. And you can see there in 2009, there was a first controlled study in otitis media with Pseudomonas. And then in 2015, the Phagoburm trial was published. And then finally in 2017, Chip Schooley, published the work by Tom Patterson and Stephanie Strathney, who treated Tom with a pseudocystic abscess with both IV and intralesional abscess phage against Acinetobacter baumannii, and that really caused, because it really worked well, and it caused a lot of activity to happen after that. This was published in 2019. Since then, a lot has happened, and I can't go through everything, but in 2000, just a few things that are relevant to NTM. Graham Hatfield published the first case of IV phage therapy, and I'll go through that case. It's a 15-year-old girl. And then they subsequently, in 2023, published a case series of 20 patients with NTM, most of which were M. abscessus. And then the other thing I just mentioned here, this is in 2025, we plan, I should say, to publish. It's going to be published at ID Week and in Europe, the joint meeting, a double-blind placebo-controlled trial looking at diabetic foot osteomyelitis. And this was probably the first one that definitively demonstrated efficacy with phage therapy plus standard of care versus standard of care alone. So I'll get into some of the information on that also. So the next slide basically just describes what a bacteriophage is. And this EM is from Graham's presentation. And they're viruses, and they specifically infect bacteria. And they don't infect mammalian hosts, is the bottom line. And the more important thing is that they're very specific to not only the genus and the species, but almost at the strain level. So if you go kingdom, phylum, class, order, family, genus, species, even below that is where they're very specific, and I'll get into why that's important in the next slide. They're prevalent on Earth. There are 10 to the 31 phage. It's the most prevalent life form on Earth, more than bacteria. For every bacteria on the surface of the Earth, there's 10 bacteriophage. They're everywhere. They're in your gut. They're in the soil. They're in hot springs. They're in glaciers. Every extreme environment you look, you're going to be able to find them. The way that they're classified is multifold. They're classified basically by genotype. Most are double-stranded DNA morphology. So you see these are ones that have tails, but there are others that are without tails. They classify them also into what they call lytic and lysogenic. So this essentially means that if they're lysogenic, they actually can integrate into the chromosome of the bacteria. And actually, that's a bit of a concern in that you can actually acquire antibiotic resistance genes and or even toxin genes. So that's something the FDA requires companies to eliminate. So we only administer lytic phage in the clinic. Next slide. Okay, so this is just a table by Dr. Altamirano back in 2019. And as I alluded to earlier, the specificity is something I wanted to focus on. It's an advantage and a disadvantage in that it's an advantage because the phage do not kill other microbiota. So it's not going to mess up your gut flora. The disadvantage is that you really need to know what the bug is beforehand in order to do the activity, in order to get you a product that's going to be active. So you need to know, number one, what species it is. Is it Staphylococcus aureus? Is it Acinetobacter, or in our case, is it a mycobacteria? And you even need to know it at the species level to really either use the phage library approach or to actually isolate and hunt phage from the environment. So that's a disadvantage. And that's why it's important to sort of initially focus on chronic infections. They're self-limited is the advantage. You know, they infect the bacterial host, and then once the bacteria is gone, they're gone. So that's an advantage because you don't have to worry about the pharmacokinetics as much as you do with antibiotics. You don't have to worry about allergies and safety. Again, they don't infect mammalian cells, so they're generally considered pretty safe. You can actually, you can actually potentially get efficacy with relatively low doses. That's something that needs to be worked out. But the idea is that if once you've infected, if you get the phage in the right place at the right time, it self-replicates and you don't have to initially administer large amounts. So that's an advantage. The other thing is, in addition to activity against antimicrobial resistant pathogens, they also have antibiofilm activity. So they have enzymes on their tails that can potentially penetrate into very difficult areas where antibiotics can't get into. So there's a number of advantages of phage therapy. Disadvantages here, they do induce phage neutralizing antibody. The clinical relevance to that is not known, however. Even if you detect it, it's not known if it matters. So that's still something that needs to be evaluated in clinical trials. There's a significant decrease in the body of evidence for phage versus antibiotics, but that's increasing as I've explained. Then also, the FDA is getting really good at regulating these and at least in the expanded access and the early clinical trials. But there is less of a regulatory framework for bacteriophages compared to antibiotics. Okay. So the next slide really kind of outlines when you would use phage, and as alluded to by Johan, difficult to treat infections is one area, and antibiotic resistant bacteria, biofilm type of infections, challenging sites of infections, foreign bodies, implantable devices, prosthetic joint infections have been treated successfully. And then the main one here is chronic infections. So that's important for two reasons. One is that it allows, these are often associated with biofilm, and that's where phage potentially has a major advantage. And also it allows, in chronic infection, allows you time to actually produce a bespoke product. So one that is actually a phage that is actually lytic against a given patient's isolate. So we're targeting, our target is to get a produced phage very rapidly, but at the same time, it's not like just pulling it off the shelf. And so in order to get that very specific phage, it does allow you to treat chronic infections, allows the time to do that. And so my conclusion is that NTMs may be uniquely suited to phage therapy, given that they're chronic and abscessus, and then NTMs are probably a first step, since abscessus is a relative rapid grower among the NTMs. And so that's where our focus is going to be as a commercial entity. And then the next slide is just showing the different treatment approaches that we use. And this is adapted from Kira, her NTM talk. It's really no different. We're focusing on inhaled here, but other people, including myself, have used all these different routes: topical for the DFO trial, intravenous injection for Tom Patterson's case, even orally by mouth is used by the Aliavi Use Institute off-label. So the other concept is what types of approaches are different companies using in terms of developing their products. So some companies are doing what they call a fixed cocktail, which is essentially just using a product that's just like antibiotics, where you don't even check the antibiotic susceptibility. You don't even know what ID you have. You don't even know what bacteria you have. You just know that it's staph potentially, or not even, you just know the genus, and you just treat patients with enough phage that have a broad enough activity that you hope that you'll be able to actually kill what's there. And there's some risk to that approach in that your phage cocktail may not be broad enough. So the other approach is a phage library, where you actually bank large amounts of different phage in hopes that when you test and treat that patient's isolate, you'll be able to identify one that works. And then the last one is bespoke phage, and that's our approach, dirt to vial. And not to say that we exclude the other approaches, because we can do them, but we actually go out and look in the environment to get to screen environmental samples for each patient isolate that comes in. And then sequential treatment can be done. It essentially is changing treatment over time, and so we can do that too. If the bacteria changes, we should be able to change in time, particularly for chronic infections. Okay. So the next slide summarizes the plaque assay. It's pretty simple. You take environmental samples, you add some liquid, you filter, and you add some liquid to a culture of bacteria, and you pour the agar in a petri dish and you incubate. And you get these plaques here, and essentially each plaque is made up of originally a source from a single particle, and can contain up to 10 to the 8th phage particles, because the amplification occurs in each one of these areas. And that's how you actually identify actually what they call phage hunting. This is the phage hunting approach. Next slide. This slide essentially is a summary of the Phages program. This is a program that is present in 150 U.S. institutions. Graham Hatfield put this slide together for his talk, and I thought I adapted it a little bit here. It's a nice picture of what it takes to actually produce a phage. And so essentially you isolate like I described here, you purify, and then once you've got the plaques, you amplify, so you reproduce large amounts, and then you need to purify the product, get rid of all the wholesale DNA and wholesale proteins. You do a DNA analysis, you do DNA sequences, you annotate to look for any endotoxins, et cetera. And then you do your comparative analysis to make sure that it's not lysogenic. And so that's really the process for producing and manufacturing a phage, just like the undergrads use in this program. And Graham actually started this program back in 2008. And it was started by Howard Hughes Institute and University of Pittsburgh, as well as a number of other academic institutions. One of the differences between the focus for this program as well as the Hatfield lab, I understand, is that they use a non-pathogenic strain of mycobacteria called Mycobacterium smegmatis, and that's because you don't want undergrads working with pathogenic organisms. But the limitation in that approach is it may take actually a lot more. It may be much more difficult to hunt than if you actually use the patient's isolate. And so that's the difference, is we actually hunt and amplify on the patient's isolate. We can amplify on other things, and we can hunt on other things, but that's our focus. So that's the difference between the two approaches. Next, I'd like to go through some of the safety and efficacy data that has been generated. The first slide on the next one is just a summary of a review, essentially a systematic review of all the studies that were conducted as the time of this publication was 2019, so it's a bit outdated now. But at that time, there were over 1,000 patients and 30 studies that had been published, and this was focused on the escape pathogens, not on mycobacteria. And escape pathogens are considered to be difficult to treat also, and multi-drug resistant. So Ephesium, Staph aureus, Klebsiella, Acinetobacter, Pseudomonas, and E. coli. They looked at they observed that people used a variety of different routes. It kind of is the whole spectrum: topical, IV, oral, inhalation, and then combinations. 87% of the studies had either decreasing or halting bacterial growth with a successful clinical outcome. 20 studies had no side effects. 8 studies had insufficient or information to make any conclusions about safety. And then there were 2 that had some side effects, but not definitely related to, didn't describe whether they were related to phage or not. And then emerging resistance was reported for 12 phage of the 34 cocktails used across all studies. And so this is sort of just a snapshot of what's been done. What you really need are randomized, double-blind, placebo-controlled studies, and one of which is the one that I started, that I designed on the next slide, basically showing, a significant improvement when you compare the placebo versus phage therapy, both in combination with standard of care. And so this is the kind of data we need in NTMs as well as Mycobacterium abscessus. When you look at M. abscessus in the next slide, I mentioned that we could treat Graham Hatfield first reported this 15-year-old girl, cystic fibrosis. This was the first case of M. abscessus being treated. And as you can see from the CT PET scan, significant decrease pre and post treatment in the lesions in the as well as in the skin. And so this girl had disseminated Mycobacterium abscessus, which was dramatically improved without any adverse reactions with a 3-phage cocktail that was administered both topically and intravenously. And then they came back in 2023 in the next slide with their case series. So this is not a randomized trial, but a case series of 20 patients, 17 of which were M. abscessus. The other ones were either BCG, Mycobacterium avium, or Chelonia. And they either administered either intravenously aerosol or both. And they showed clinical improvement or microbiologic responses in 11 of the 20 patients. They did, again, they did look at neutralizing antibodies in that study also. Again, that's still TBD as to whether that means anything. And then finally, the next slide is just showing we have treated 1 patient at TOCA, and we did also show promising microbiologic response. We are screening currently 7 more patients and have found phage for 4 of them. So we don't rule out finding phage for the others, but we're having a pretty good hit rate so far compared to what's been done in the past. And we'd like to expand our compassionate use program even more. The next slide sort of describes what kind of expanded access programs exist. On the lower right there's a single patient expanded access IND, which is what's typically used. There's also, FDA has described what they call intermediate population IND, which is hundreds of patients where it's all sponsored by the company. Single patient is typically, those are managed and sponsored by the individual physician. And then you have a treatment protocol, which also is expanded access, but that happens in between submitting your application and getting approval. So those are thousands potentially of patients who may need a product that's life-threatening, who have a condition that's life-threatening. And so where we fit in is there on the left there. We would like to collect sputum from as many patients that have both M. abscessus, but even NTMs, and we can isolate phage. We can hunt phage, and we can culture bacteria from sputum, or actually get the culture itself, and then put it through the process of hunting and manufacturing and amplification, as I described earlier, and then either set up a single patient IND, or at some point in the future, once it's IRB approved, we would do an intermediate population IND. So that's our plan. In conclusion, the phage have been around forever and billions of years. They're the natural enemies of bacteria. there's a well-deserved rebirth in the treatment of with phage, given the challenges in cost and tolerability of antibiotics, biofilm, chronic infections. Very few rigorous clinical trials have been completed, but there have been few that are encouraging, as I've described. We think that M. abscessus infections may be the best initial focus, both because it's a relatively fast grower and it's associated with a fairly chronic infection, which allows us time to manufacture. We have the ability to actually, use a phage library, or more importantly, use what I call dirt to vial approach, which is actually isolating a phage from a patient's from the environment, using the patient isolate for both hunting and manufacturing. We have a bacterial bank of over 150 abscessus isolates. We're actually screening for in 7 patients, and we're slowly developing. One of my remits is to develop a physician network who are interested in in treating patients with M. abscessus. So even if you don't have a physician that has active interest, we can probably find one, because we have a pretty broad network across the U.S. All right, that's all I had to say. I think there should still be time for questions. Okay.
I think I have
I'm sorry.
a slide or
Johan.
two, actually.
Yeah,
Okay, great.
Yeah.
Away.
Yeah. So great. Thank you, Bob. So what we're doing right now, what we're planning to do in the near future, we'll maintain the expanded access program for M. abscessus patients, and this is to make treatment accessible before we kick off clinical trials. Then we want to conduct the clinical trials 1 through 3 to license M. abscessus so everybody can get access. We will focus on the nebulized route and then expand to IV if needed. We want to build the world's largest repository of M. abscessus strains, phages, and data, ideally compatible with existing or planned patient registries. We want to get licensure of M. abscessus phage treatments in the US, Japan, Europe, etc., or places like Australia as well, and then move on to other non-tuberculous mycobacteria and then other chronic infections. So one thing that we're planning to do very soon is open up our biobank for other people to join, and specifically patients. So we're planning an M. abscessus strain biobank. This would be the first step for eligibility for future expanded access treatment and clinical trials. It might help you, if you're listening to this and you have M. abscessus right now, in the future, but it may also help other people in the future if you donate a sample and we are able to develop a therapy that can help you or can help other people. Now, we are not quite ready to open this up yet, but if you are interested, here is a QR code where you can sign up for updates, or you can go to tolka.ai/updates, enter your email address, and then we will let you know as soon as we have everything ready to go. I guess I can leave this slide maybe, and then now we can move to questions.
Okay, terrific. I'll get started with the questions. Johan, somebody's asking if you had done phage therapy yourself, and I think you had. I'm not sure if you had addressed that at the beginning.
So yes, I am patient number 1. We have not published anything around this yet, but eventually we will.
Are the services of your company covered by any medical insurances or Medicare?
So for expanded access, this is all free. It's going to be free for the expanded access program and clinical trials as well.
Yeah, the delivery of the product is free. The physician, administration and etc. That would not be free, but yeah.
Okay. We have some questions about other co-infections. Somebody's asking, do you do phage research for Pseudomonas?
Not yet.
Yeah, that would fit into the other chronic infections. I know that like the phagoburn and the OTI, the UTI, and I've done Pseudomonas phage, but that's not the top of our list. The reason, again, we're focusing on abscessus is because, number one, we're small, we need to focus. but also it's going to give, it's even associated with even more chronic infections. And so it allows us, for multiple reasons, allows us to get commercialized sooner.
Okay, so here's a couple of follow-on questions about that, and I don't know if you have any of these answers, but I think it's probably in terms of information worth exploring. Can you treat NTM and Pseudomonas with phages at the same time? Like, could you do a cocktail for both, I guess? Or would you want to do that or not?
You could, yes, you could. And it's the challenge is really just in terms of bandwidth. For example, I was involved in helping the NIH and the ARLG group develop the Pseudomonas phage for CF. And that was a cocktail approach, and that was IV. And other companies have looked at nebulized. So I mean, that's being done for Pseudomonas, and you can combine. People have combined phages from different. In fact, that's what's been done historically at Oliava, in Georgia, not for invasive infections, but in theory you can, but that's probably not the first thing we want to do. We need to focus.
Can phage therapy be effective for a bronchiectasis patient and bacterial infections that are colonized and that are not NTM? So I guess, like, how effective can it be?
So one question, I think that's a big question. It's one I'm very interested in. It's if let's say you've got a patient with M. abscessus, and it's their first they're colonized, right? And they don't have significant symptoms. Is there an advantage to treating them just to get rid of the colonization so that they don't develop symptoms later, right? I would think there would be, but you'd have to demonstrate that you could actually clear it up. And there's a challenge in terms of the compassionate use, and that typically that's done with more severe infections, people that have life-threatening conditions, et cetera. And so that might be best done in a phase 1 trial, where you take anyone who has a positive infection but aren't dramatically symptomatic, and look to see if you clear the infection. But that would be the endpoint. And the surrogate to that would be, you would hope that would reduce long-term complications, but you'd have to follow that up to demonstrate it.
Okay. Somebody's asking, do you have to repeat the phage treatment with each new flare-up or infection in chronic infections?
So Johan had spoken to that in terms of, and I had spoken to it in terms of quote unquote sequential treatment. So you can, and if you're having exacerbations, and if you get good clearance, let's say that you give your phage therapy and you get good clearance, and maybe some improvement in your patient-reported outcomes, decreased cough, better activity, feeling better, less fatigue, and then you have an exacerbation, it does make sense to take another bacterial isolate and look and do the testing. So you can test what they call phage susceptibility testing, sometimes referred to as PST test, just like antibiotic susceptibility testing, AST, and you can test the new isolate against the phage that is being used to determine whether it's still lytic. And if it's changed, then we have the capacity to provide a second bespoke phage. And that's one of the advantages of our program, is that we can adjust as time goes on, particularly for patients with a chronic infection. So that's a good point, something we're very focused on.
Okay. How would a patient be able to submit a sputum sample to your company to help the database grow?
Let me just make one point. I mean, it needs to be IRB approved. So once we get our biobank IRB approved, we'll reach out. But at this point, we can't do anything unless we'd like to get IRB approval. for even a biorepository. And so once that's done, we can send out a note. If you put in your email address using the QR code, we can reach out to anyone that's interested. For Johan,
yeah, or you can type this into a web browser, and Bob is absolutely right. We are waiting for IRB approval probably in the next 2 months or so, and then we can give you more details on what this process will look like.
Okay. Would phage work for NTM if somebody's having negative sputums because they've been treated?
Well, that is a very interesting question. We've found that sometimes some assays are more sensitive than others. So a negative sputum, it can test negative, but with a different assay, it can test positive. It probably just means you have a very low amount of NTM rather than nothing. So this all ties back to why we wish there was more data for each patient, right? If we'd really had great data to show that it's really gone and we had multiple data points that were very sensitive, then you probably shouldn't treat with phage therapy. But it's very, very hard to get that quality data at the moment.
My sputum sample for abscessus was sent, but my doctor told me that it didn't find a match. Does this mean that I don't have a chance for phage treatment for the rest of my life?
So, go ahead.
So we will keep searching until we find a phage. So it's very possible that you might not have a match today, but in a few months we might be able to find one. So the earlier we get the sample, the better chance we have of finding a match eventually.
Okay. So, all right. Is hearing loss alone a significant criterion for qualifying for phage therapy despite antibiotic sensitivity?
Yeah, I mean, I don't think I probably can't answer that without knowing much more detail about the patient. But certainly, if you're required aminoglycosides based on your AST profile, and you've had hearing loss as a result of the aminoglycosides, I would think that you would qualify. I don't think the FDA, MSSS is a bad bug, and so I'm hoping that the FDA would agree with at least our opinion that there should be a fairly low bar. Getting that alone, having MSSS alone is not a good predictor. So I would think you would, but I can't say that definitively. I don't know, Johan, if you have anything else to comment
on. So the official FDA guideline says it needs to be either severe or life-threatening, and you need to have tried existing therapies. Severe is really up to the treating physician to decide. So it does not have to be immediately life-threatening. And I would think that many NTM infections would qualify as severe. And it is also common that patients have tried the existing drugs as well. So it is relatively common, both of these things. But yeah, it's really in the eyes of the physician what counts as severe initially. And then they send that proposal to the FDA. And the FDA have been great so far. Our interactions with the FDA have been really good. So they've been very professional so far.
Okay. Do phages always have to be used in conjunction with antibiotics?
They don't have to be, but that's generally what's been done. I think most researchers and most experts would agree that phage should be added to existing treatment. Over time, I mean, I would love to be able to say you can prevent, IV treatment by using oral and nebulized treatment for, NTMs. But, if you require IV treatment and it's still not working, you could potentially try stopping the IV treatment after you started phage if markers of infection has decreased, but it depends on the patient. In general, though, you want to keep standard of care and then add phage to it. Would be my general thought.
Yeah, I should add that much of this is speculative until we have clinical trial results, and that's why we're working as hard as we can towards clinical trials. That's going to be the definitive answer.
Speaking of clinical trials, we have some people from outside the US who wanted to know if they're coming from the UK or Australia, if they were willing to travel to the US for treatment, would it be accessible to them, either on compassionate use or eventually for clinical trials?
So if you are in Australia, there is a organization called Phage Australia that you can contact, and you can actually get phage therapy in Australia under a very similar framework as the expanded access framework here in the US. We are in active discussions with them about screening phages for Phage Australia. So that would be the best way if you're in Australia. In the UK, we have a few connections in the UK as well, but we haven't done any screening for any patients in the UK so far.
Okay. One more follow-on question on that is, has any progress been made in Georgia or Eastern Europe for NTM applications?
I'm not aware of any. I don't know, but I'm not aware of anything that's been published. But I'd have to look.
My understanding, and this is a few years ago, but a couple of years ago, they were not doing NTM in Georgia. So I don't know if that has changed.
Has been more on, historically their focus is more on topical, oral, phage, not even IV, maybe some nebulized, but it hasn't been on, and part of that is, the manufacturing process is required to give systemic phage, at least by the FDA and EU, is high. And so some of it's even over the counter. Some of their cocktails are even over the counter historically.
Okay. Somebody asking about phage MAC treatment. How far in the future for your company, or do you know what the general state of that effort is?
Well, so MAC is challenging because the pathogen grows very, very slowly compared to Mycobacterium abscessus. So once we have a good amount of experience with Mycobacterium abscessus, we will start looking into MAC. But yeah, it depends on the progress we're making on M. abscessus. And I think, can't speak for everybody in the world, but I would assume everybody else has similar problems with MAC.
Okay, we have two people asking about Canada. I guess if if they're from Canada, would they have access as well to this program?
So yeah, I mean, to this program, sure. I haven't reached out to any Canadian investigators yet that might be willing, but I think we'd be happy, and we've reached out to other countries, so it's not like we can't do it. And the first step of just getting it isolated is fairly simple. Once we get the biorepository, patient-based biorepository IRB approved, it's fairly simple. So, check, sign in, and once we get it approved, we can add.
Okay. So somebody's asking about, the sort of what happens after the phage therapy. Does it actually like tackle the NTM and clear it, and then after this, there's been damage to the lungs. Does it they're asking if it helps rejuvenate lung tissue, which I don't think that's actually possible.
Yeah, I think it's fair to say the best you could probably expect is it wouldn't get worse if, and there's a lot of caveats. Number one, if the majority of the bronchiectasis is due to the infection, which may not be the case, but it certainly would take out one factor. It's sort of a is the infection making the bronchiectasis worse, or is the bronchiectasis causing the susceptibility to infection, the chicken and the egg problem? And it cannot hurt. It may not help, but it cannot hurt, and that it may decrease the deterioration of your bronchiectasis. It's a may.
Okay. So another question about MAC. So I guess when you were doing your presentation, you talked a little bit about sort of the overall process for this. Would this process work the same way for MAC as well? And if somebody was taking this into the future, or if you decided to do that in the future, would it be a similar process?
Yeah, I think it would be. I mean, we would probably, again, get some experience at hunting, amplifying, and manufacturing the NTMs that are not M. abscessus as a second effort. And that would initially be just providing material for compassionate use. And then there would be, if there's interest from an investor perspective, we would go ahead and develop it for commercialization. So, and then once it's licensed, of course, it's licensed and it's available. But again, there is no phage license at this point. There are programs that are in phase 2, but nothing licensable.
Okay. Somebody's asking about Trikafta. If you're taking Trikafta, could you have phage therapy while you're on Trikafta?
Yeah, I don't think there's any reason to say you don't need it. Certainly there are less risk for infection, but if they've got symptoms that are severe, life-threatening in particular, if they've got M. abscessus, despite being on Trikafta, they can take phage therapy.
Okay. Somebody's got a multi-part question that sort of they're asking about like sort of what happens after the phages deal with the infection. Once the NTM is eliminated by phages, what happens to the remaining phages? If they're, do the targeted NTMs become resistant to phages? And the phages that are introduced into the lungs, do they remain in the lungs or do they migrate to other areas of the body?
So the phage, one thing that's important to remember is the phage cannot survive unless it has a host. So once the host goes down to a level where they're gone, if they're zero, then there is no more phage because they cannot survive unless they replicate in a host. So that's the beautiful thing about the phage, is that they continue to replicate until all the bacteria are gone. And so hopefully that answers the question.
I can add a little bit to that. So already today, everybody has some small amount of phage circulating in their bloodstream even, and the body is constantly filtering out these phages through the liver. So it's a very common process in the body.
And they were asking sort of about the targeted NTMs. Do they develop like a resistance to the phages?
These are all great questions.
That need to be answered.
Your clinical trials will be the ultimate answer to these questions. Whether
Okay.
you get,
Here's an
a gold standard clinical trial.
Yeah. Do phages have any difficulty penetrating the waxy outer membrane of NTMs?
Also a great research question. I think clearly there are phages that can do it. But yeah, it may very well be that it's harder to find phage for NTM because of the cell wall. It seems to be harder to find NTM phages in general compared to other phages. But these are all open research questions that we have many years of research to figure out.
So somebody's asking, why do we use antibiotics, or why do we like to use antibiotics in conjunction with the phage therapy?
Well, number one, they've got, at least in the Western medicine, they've gotten years and decades of experience. So, and they're licensed, they're approved by FDA, they're recommended by IDSA and other guidance documents. So there are multiple reasons to use them as sort of the standard, and they are considered standard of care. Phage are investigational still, so you can't just buy them. You have to get them through some alternative means, either through compassionate use and eventually through clinical trials. And then once they're licensed, they'll be more likely to be available and used. But that's why.
Okay. Somebody's asking an interesting question. Is there a document that a layperson can read to better understand the phage process? I don't know that there is one, but maybe we should actually work on one and put it up like on our website.
Yeah, I mean,
Yeah,
well,
that's a
you have your first presentation. The first thing I did was listen to Graham Hatfield's and Kira Cohen's presentation, which I couldn't improve upon it, so I used some of their slides. So I just adapted them. So that's a good place to start. Listen to your first NTM lecture. It goes into more detail.
Okay. So somebody's asking specifically about cavitary NTM infections. They want to know how well phage treats it. Is there a differential between, cavitary and non-cavitary, or is it really dependent on the strain?
Yeah, I don't think anyone knows the answer to that. I mean, ideally, that's part of the reason I think I like the idea of going with a nebulized treatment initially, focusing on nebulized treatment, thinking that you'll get into the bronchial tree and at some point it may penetrate, the cavity. Alternative, it may be that you do need IV treatment for cavities, and it just doesn't get in. So you need to get the phage to where the bacteria are, though they're not going to have an impact. And how to do that is still, TBD. One thing that hasn't been well investigated, we haven't had 40 years of development with phage, so we don't know what is the right frequency, what is the right dose, although that's less of an important question. What is the right route, right? And so those are things that need to be, addressed in the clinical trials.
Right. Do you have any insights as to why a faster growing mycobacteria seems to be easier to match a phage? Like why abscessus versus MAC? Is it just that we're finding more of those phages and we don't know where to look for the MAC phages, or?
Do you want to take that one, or I can attempt?
I mean, it is largely a scientific question. It's hard enough finding phages for M. abscessus, and we know that MAC would be even harder just because of the slow growth. Biology can be really complicated sometimes.
Yeah,
I don't know if you have anything to add to that, Bob.
Well, I think it's just, we could ask our manufacturing folks, but if it takes 2 weeks to grow, that slows the process down dramatically. And just from a commercialization perspective, if you want to get something approved quickly, you can do it 5 times faster if you focus on M. abscessus. If we had unlimited resources, we would do everything at once. But there isn't unlimited resources, so we're forced to focus. So it's not that it can't be done; it's just that there aren't enough resources to do everything at once, and it would take longer.
Okay. Last question. If you're comfortable sharing, Johan, what has been your outcome after using phage therapy?
Yeah. So we will definitely share more information about this in the future when we're ready to publish more of it. We've been encouraged by the microbiological data coming out of that. So we'll have more information to share shortly.
Okay, so you'll be publishing. Okay, well, thank you. All right, so this has been very enlightening, and I think we got through all the questions that we had. I'd like to thank you both for for taking the time today. This has been terrific. It's always good to hear an update about, the progress of industry working in this area. I think there's been a lot of fascination with this topic over the past several years. So thank you again. And everybody, thank you for joining us. And please don't forget to sign up for the webinar on August 28th with Ann O'Donnell. Johan, Bob, thank you again. And we will talk soon.
Thank
Thanks,
you.
everybody.
Thank you, Amy.
Have a
Thank
great
you,
day.
everybody.